Hi everyone! Today we're going to talk about gel electrophoresis. Now, gel electrophoresis is actually this way that we take charged particles and get them move in response to electrical force. So basically we know that positive things will move towards something negative and negative things will move towards something positive. Now, we use this to take pieces of DNA that have been cut up by a restriction enzyme and spread them out. Why would we bother doing this? Well, basically every single person has different DNA. So if we cut up our DNA, it will end up in different sized pieces. And when we spread those out, they make what's called a DNA fingerprint. And in crime scenes, they can actually take samples of DNA found at the scene and spread them out and use them to compare different possible individuals who could be the criminal. This is also used outside of crime scenes. For example, in ecology, zoos will make a lot of species diversity plans. If they want to increase variation in a species of a particular kind of organism they have, they're going to talk to other zoos. They're going to check, let's say we're going to look at polar bears. If we want to mate these different polar bears, we want the least similar polar bears, and we're going to pick those two to mate. So they're going to cut up the DNA, compare them in these gels, and find the most different ones, and those are the two that they'll say, okay, this will cross these two and try to have some babies at the zoo. Now, in order to do gel electrophoresis, the first thing you're going to need to do is make a gel. And there's this apparatus that has kind of a comb in it right here, and then it has this empty frame. And what you do is we melt agarose gel, and we're going to do it in the microwave, and we pour it into this mold. Then you're going to put this comb in right here. It's kind of nice because then it'll cool and solidify, and then you'll pull the comb out and it'll leave some wells that we'll talk about later. Now, why agarose? Agarose is taken from seaweed. It has a uniform pore size, and that means if DNA is moving through these pores, no matter where they're at in the agarose, if it's the same size, it should move at a similar rate. And also, what's nice about agarose is when we melt it and pour it into a tray, it's sort of like a thick jello. You can pick it up, but it's a nice usable form. Now, the second thing we have to do in gel electrophoresis is put it into an electrophoresis chamber. This, you're also going to add a liquid buffer, and that's going to be poured in here and completely cover it. You'll notice that there's also some electrical charges on either end that will go in here. So you're going to lay your gels inside this liquid and we can run the electrical charge through it. The next thing you're going to do is add prepared DNA samples. Remember how I told you we were going to pull that comb out? When we pull that comb out, it leaves behind these wells. We're going to use micropipettes to deposit small amounts of DNA into each well, and they'll make sort of like a column. So you'll put one sample into this position and it'll run this direction, one sample into this position, and another into this one. Now, also, at this point in time, the DNA is invisible, so we add some dye to it to make sure we can see it, just when we're dropping it into the gels. And again, like I said, we're going to put the dye into these wells and the DNA once the combs have formed those wells. Now, the next thing we need to do is hook it up to a power supply and turn on the electricity. So one of the things you want to pay attention to is the location of the electrodes. Notice how this is a negative powered electrode there. We've got a positive electrode right over here. And think for a second, what kind of charge does DNA have? Think about that for yourself. I'm hoping you thought that DNA was negatively charged. And so I want you to pay close attention and make sure that when you place your gels in, the gels are close to the negative side. The reason is when we put the negatively charged DNA in, we want the DNA to run in this direction towards the positive electrode. Now, DNA is going to move differently on a few different things. The more electricity, the faster it's going to move. So if you don't have a lot of time, you're going to have to dial up that electricity in a class period. Also, smaller pieces of DNA will travel faster than larger pieces of DNA. I want you to think about that, and ask yourself, why would that be true? I have a little example here to help you see this. So here we have our negative electrode and our negative piece of DNA, and we've got our positive electrodes. So the DNA, of course, is going to move from towards the positive electrode because it's negatively charged right there and there. I have a very large piece of DNA that has to pass through uniform agarose pores, and I have a very small piece of DNA. Which one moves more quickly through these pores? Of course, the smaller pieces of DNA can move more quickly. That becomes very important in gel electrophoresis. Now, when we're doing this, what exactly are we seeing? Well, we see the dye moving, not the actual DNA. So the dye helps us as we're going through the procedure, but it still doesn't help us see the pieces of DNA, and we have to do something to actually see it. So we add once we have used the dye to watch that it's followed with the electricity and we remove the gel, we're going to then stain the DNA. This is different from the dye. I want to emphasize that. And once we've done that, we get something that looks like this. Now, if you've got a light box behind it, it might glow depending on the kind of stain you use, or it might simply look blue depending on the kind of dye you used. So here's an example of a gel after it's been run. You can see here is our crime scene sample. These are our different possible candidates or potential criminals, our suspects, and we are going to compare their bands of DNA to the criminal DNA, the stain from the crime scene. Now, what you're going to do is you're going to look at this and tell me which one of these individuals is the person who left the blood at the crime scene. I'm hoping, since you recognize that each band represents a fragment of DNA, and that every person has different DNA, that the only person who could possibly match the blood stain would be the actual person who left the blood. So if we look, I'm hoping you noticed it was John. And also I want you to think about the fact that each band represents a piece of DNA. Where will the smallest pieces of DNA be located? I'm hoping that you notice that the smaller bands were down here towards the bottom. Again, because smaller pieces are going to move more quickly, and the larger pieces should be up towards the top, and they would move more slowly. So if these bands match up, John with the blood stain, we are very confident that this blood stain came from John. Now, I can't guarantee that John actually performed any sort of crime, but I can say that it is almost 100% certain that that is John's blood at the crime scene. Now, restriction enzymes. One of the questions you might ask is, how did we get that DNA cut up into so many pieces? Well, we have these lovely restriction enzymes that we've stolen from bacteria, and they are very equipped to cut up DNA molecules. Now, enzymes will cut in very specific sequences, and that so it's going to, every single time it sees that sequence in DNA, that is where it's going to cut. So, for example, let's say we chose a restriction enzyme that cuts DNA whenever it sees ATTG. So, if this was one of our strands of DNA, every time there was an ATTG, so like there, it's going to cut at that specific location. Now, again, these are not actual scissors. This is enzymatic cutting. But for the purposes of this PowerPoint, we've got some nice scissors here showing their cut. Now, this 2 cuts ends up giving us 3 pieces of DNA right here of different sizes. Now, if we take those pieces of DNA and they've now been cut up, and we go ahead and we load them into one well of the gel electrophoresis. Notice again, they've been loaded at the negative electrode end, so because they are also negatively charged and will travel towards the positive electrode end. And I want you to think about which pieces will move more quickly through the agarose gel. And we're going to go ahead and we're going to turn on the electricity and let it go. Oh, and then we'll stop the electricity. Notice I stopped the electricity not when everything had run off the end, But instead, when the little pieces had moved all the way to the edge and the big pieces of DNA were still sort of spread out, we don't want to let all the DNA run off. We like it to be nicely spread out. How do I actually determine when to turn off the electricity? Well, that's what the dye is for. The dye will move along at the front here, and we don't want the dye to completely run off the end of the gel. Now, so at this point in time, you won't see a fat band right here, or a medium-sized band right here, or a small band right here. Instead, you will see the same size band. The only way you'll know they're different sizes is because of how far they have traveled along the gel. I hope this helps.